So for part 2 of the yeast respiration lab, we'll be collecting data. We'll record that data in our data table here. Notice that you have observations on the left, what does it look like, and circumference of the balloon on the right, how much carbon dioxide was produced. So remember, we have 2 test tubes. We have A, our control, and B, our experimental. The first thing we're going to do is notice any colors or bubbliness for each of the test tubes and write down what they look like in comparison to one another. Then we're going to take our string, so every group should have a piece of string and a ruler, and we're going to be measuring the widest part around the balloon, and if there's no air in it, we're essentially just measuring the width of that balloon, the circumference of that balloon. So we're going to take our string, wrap it all the way around the widest part. Okay. So we might need 2 hands for this. And once you think you have the widest part like this, you're going to kind of hold your finger at the end of the string, and then we're going to be measuring how long that piece of string is. So for me, it's about 13 centimeters, and we're going to record in centimeters. Then I'm going to do the same thing with my other balloon and measure all the way around my circumference. Keep my finger on it and measure that distance. And for mine, it's about 20. So I'll record those things in the data table. And really important, in order to clean up, we're going to actually take the balloon and rubber band off, throw that in the trash. We're going to rinse really, really well for each test tube and then place those on the drying rack when you're finished.